Hi everyone, welcome back to the second half of chapter 12. So we'll be talking about part 2 of chapter 12 today, and it's very short. So it won't be too long before we're done with chapter 12. And before we move on, I would have to suggest watching all the animations again, including the crash course videos. So the crash course video for action potentials is the material we covered last time. There's also another crash course on synapses that's relevant for this section. So just starting off with a fun fact, how many neurons are in the brain? So we said there are about 1 trillion neurons in the nervous system, and we have about 100 billion neurons in the brain. And each of those neurons can have a connection with one more neuron or more. Some neurons can have up to 1,000 of these connections. So each connection is called a synapse. It's where they almost touch. It's where they connect. And within the brain, there are between 500 trillion and 1 quadrillion neuron-to-neuron connections. So there are up to 1 quadrillion synapses in the human brain. And we spoke about neurons being able to receive, process, and send information. So we gave an example of, let's say we're pricking ourselves, or we touch something that's hot, we touch a hot surface. So what happens is the sensory receptor in our finger is communicating with the sensory neuron, which is then communicating with an interneuron in the CNS. Then the interneuron is making a decision and then communicating with this motor neuron, and the motor neuron will then communicate with this muscle cell. So these are all synapses. These are synapses in between the connections between the neurons. So the synapse is where a neuron almost contacts the next cell. And the terminal bouton of the presynaptic neuron is almost contacts a postsynaptic cell. So let's look at this example specifically. Here is the sending cell. This cell is sending a message. So an action potential is traveling along this axon. And since this neuron is sending something, it's sending the message, we call this the presynaptic cell. It's pre-synapse. It's before the synapse. Once the action potential reaches the axon terminal, it reaches the terminal bouton. And that's closest to the synapse, which is the space between the terminal bouton and the dendrites of the postsynaptic cell. So this cell over here is receiving a message. So we call this the postsynaptic cell. So the message is going from this cell down the axon to the terminal bouton. Something's going to happen across the synapse that sends the message to the dendrites of the postsynaptic cell. And then, of course, this could then send the message along this axon, and this becomes then the presynaptic cell if there's another cell to the right over here. And this synaptic cleft, right, the space is around 20 nanometers wide. So there's a very, very tiny space. The neurons don't physically touch each other. So as we will now see, in order to propagate or to inhibit action potentials from neuron to neuron, we need to use neurotransmitters. So chemical messages and electrical messages are being sent in the brain and the spinal cord and throughout all of our nerves. So neurotransmitters are part of the chemical signals that our nervous system uses. So action potentials are more like electricity, where neurotransmitters are more like chemicals. So let's zoom in over here. This is a presynaptic neuron, and then this is a postsynaptic neuron or a postsynaptic cell over here. This could be a muscle cell, this could be another neuron. And this is the synaptic cleft. This is the space in between the presynaptic neuron and the postsynaptic cell. So over here, we see that when the action potential reaches the end of this axon of the presynaptic cell, we're going to see that the terminal bouton, which is this little protrusion over here, the terminal bouton will release neurotransmitters into the synaptic cleft. So over here, this is the synaptic cleft, And this is the membrane of the postsynaptic cell over here. That's what's being zoomed in over here, is this portion. So when the action potential reaches the end of the axon, the terminal bouton will release neurotransmitters into the synaptic cleft. Neurotransmitters will then bind to receptors on the postsynaptic membrane. This is the postsynaptic membrane in purple here. This is zoomed in. And here is the green neurotransmitter binding to this receptor protein on the postsynaptic cell membrane. And what this neurotransmitter is going to do is change the postsynaptic cell that will either allow it to stimulate more action potentials or inhibit more action potentials. So these neurotransmitters go from one cell, from the presynaptic neuron, to the next cell, basically saying, let's continue more action potentials or let's stop action potentials. So neurotransmitters are chemicals that either stimulate or inhibit nerve impulses in the next axon. So take a second to answer this question. So the receptors for the neurotransmitters are located on the plasma membrane of the postsynaptic neuron. The answer is C. And neurotransmitters are in the body, throughout the body, as well as exclusively in the brain. So we have some neurotransmitters that are just in the brain, some that are used throughout the body, and current research has identified over 100 different neurotransmitters. So like I mentioned, neurotransmitters can either excite or inhibit the receiving cell. And that response depends on the type of neurotransmitter used, right? The type of and the type of receptor that's available. So the type of neurotransmitters and the type of receptors. Also the amount of neurotransmitters. And finally, like I mentioned, the type of receptors on the receiving cell. So it's the type of neurotransmitters, the type of receptors on the receiving cell, and the amount of neurotransmitters will determine the response on the receiving cell. So neurotransmitters are what going to carry the message from neuron A to neuron B. Right? So neuron B has specific receptors on its cell membrane that's specific for a neurotransmitter sent by neuron A. So I'll say that one more time. Neuron A is going to release neurotransmitters. Those neurotransmitters will be released into the synapse and will then find receptors on neuron B. If neuron B receives the right chemical signals, then neuron B will make its own action potential and pass the message down to the next cell. Neurotransmitters are very short-lived chemical messages, and these are quickly broken down in the synapse. So these are very short-lived. They only allow a message to be sent to the postsynaptic cell for a very brief time. The neurotransmitters are then broken down so they can no longer send action potentials. Again, we said there are 100 neurotransmitters, but I want you to know just 4 examples for now. You'll be familiar with some of them, like serotonin is the mood neurotransmitter. Dopamine is the pleasure neurotransmitter. Adrenaline is the fight or flight neurotransmitter. GABA is known to be a calming, relaxing neurotransmitter. Acetylcholine is associated with learning. Glutamate is associated with memory. And if you look closely, all of these neurotransmitters are just chemicals that are made of carbon, hydrogen, oxygen, and nitrogen. These are all chemicals that just can bind to certain membrane proteins on other neurons or muscles and either cause an action potential or stop an action potential. So I want to talk about 3 different types of synapses. The first 2 are a little more complicated. The third is pretty easy. But I want to keep in mind what we're talking about. these neurotransmitters are interacting between two neurons, saying, do we continue sending action potentials or do we stop sending action potentials? So we'll talk about a cholinergic synapse first. And a cholinergic synapse is one that uses the neurotransmitter acetylcholine, abbreviated ACH. So here we are at a cholinergic synapse, and these are known as excitatory because these are going to propagate. These are going to excite the action potentials in the postsynaptic cell. Right? So these are excitatory. They're going to send an increase of nerve impulses. So it all starts at the synapse, and the presynaptic neuron will have a action potential. Right? So the action potential will be sent along this axon of the presynaptic neuron. And once the action potential reaches the terminal bouton of the presynaptic neuron, what's going to happen is special calcium channels are going to open up. So when the action potential reaches the end of the presynaptic neuron, calcium channels will open up and calcium will be released inside the presynaptic neuron. So the calcium channels open in response to the change in membrane potential. So remember, an action potential is changing the membrane potential. And as the action potential reaches right here, these calcium channels are going to open. Calcium is going to flood inside the terminal bouton of the presynaptic neuron. And what's going to happen is acetylcholine will be released from vesicles. So the increase in calcium in the presynaptic neuron will trigger the release of acetylcholine into the synapse. So the green acetylcholine will be released into the synapse, and the acetylcholine will diffuse across the synaptic cleft and bind to the postsynaptic receptors. So these are shown in orange here. And once acetylcholine binds to these receptors, these will allow sodium to diffuse inside of the cell. And what happens when sodium can diffuse inside of the cell, that will depolarize the postsynaptic neuron. And depolarization of the postsynaptic neuron, if there's enough depolarization to reach threshold, it will cause an action potential in the postsynaptic neuron. This will then cause a different type of sodium channel to open. These sodium channels that open first were controlled by acetylcholine. So we call those ligand-gated ion channels because it was a gate that was controlled by acetylcholine binding. If enough sodium comes inside of this postsynaptic cell to reach threshold, then voltage-gated sodium channels will open. That's not shown in this picture here. And then it will increase depolarization even more. So what has to happen is after the action potential is sent, we have to turn off stimulation, or else the postsynaptic neuron will fire indefinitely. So what happens is the presynaptic cell will stop releasing the neurotransmitter. So the ligand-gated sodium channels will stop allowing sodium in. And what will happen is the acetylcholine that's inside the synapse will be broken down by an enzyme called acetylcholinesterase that's always found in the synaptic cleft. So acetylcholine will be broken down into acetate and choline, and those fragments of the molecule will be reabsorbed into the cell for recycling. So take a second to watch this video that basically summarizes what we just learned. Another type of excitatory synapse is an adrenergic synapse. And this is one that we've spoken about before in terms of signal transduction, how cells communicate with each other. We spoke about this in the context of, I believe it was glycogen in the liver. So adrenergic synapses require a different neurotransmitter called epinephrine or norepinephrine, also called noradrenaline. So here, it's a different kind of interaction, a different kind of mechanism that uses second messengers like cyclic AMP. So here we're still at an excitatory synapse, but this postsynaptic neuron has a different type of receptor. These receptors are not ion gates, but they're transmembrane proteins called G-protein coupled receptors, or GPCRs, that are associated with a G protein on the inside of the postsynaptic cell. So here on the synapse we have, this is called a G protein coupled receptor. This neurotransmitter receptor is a G protein coupled receptor, and in purple is the actual G protein. So what happens is, the action potential will be sent down this neuron, and now norepinephrine will be released into the synapse. And over here, norepinephrine will bind to its receptor. In this case, it's actually called the adrenergic receptor. And the adrenergic receptor will change its shape when norepinephrine binds to it. And that will activate this G protein. So what we spoke about so far is step 1, action potential reaches the end of the presynaptic neuron. Norepinephrine is released into the synapse. Norepinephrine binds to its receptor, which then activates a G protein. That G protein will activate an enzyme on the membrane called adenylate cyclase. And adenylate cyclase converts ATP into the second messenger cyclic AMP. And cyclic AMP can do multiple things from here. In terms of action potentials, what cyclic AMP can do is it can open up sodium channels, allowing depolarization of the postsynaptic membrane. So in this way, we sent an action potential. We excited this postsynaptic neuron by sending an action potential along this way using not acetylcholine, but norepinephrine. And norepinephrine opened up these sodium channels indirectly. So what happened was the norepinephrine had to first tell the receptor to activate a G protein to convert ATP into cyclic AMP, and then cyclic AMP turned on the sodium channel from the inside, and then allowed sodium inside the postsynaptic neuron. So the third and final type of synapse we'll talk about is an inhibitory synapse. So this is a type of synapse that will stop the transmission and propagation of the action potential. So a GABAergic synapse employs aminobutyric acid, or GABA. GABA aminobutyric acid is what GABA stands for. So that's the neurotransmitter. And GABA is known as a calming or an inhibitory neurotransmitter because it slows down action potentials. So a nerve signal can trigger the release of GABA into the synaptic cleft. And the GABA receptors are specifically channels that allow the chloride ion in. And chloride is a negatively charged ion. So when GABA is released into the synaptic cleft and binds to their GABA receptors, those GABA receptors open up and allow chloride in. What that does, the negative chloride rushing into the cell makes the inside even more negative than resting potential. So that's going to make it even less likely for that cell to be depolarized. You're making it hyperpolarized. So the postsynaptic neuron is thus inhibited and less likely to fire. So in that way, GABA is inhibiting or slowing down the action potential. So those are how synapses work. And neurotransmitters are mimicked by drugs. So a lot of drugs work by mimicking the natural effect of neurotransmitters, or work by altering how the natural neurotransmitters bind to receptors in our synapses. So examples include stimulants like caffeine or nicotine, opioids like heroin or Oxycontin, hallucinogens, even antidepressants. So like an SSRI, which is a common type of antidepressant, works by increasing the time that serotonin is left in the synapse. So an SSRI stands for selective serotonin reuptake inhibitor. So it inhibits the reuptake of serotonin, so it stays longer in the synapse and thus sends more excitatory, happy, quote unquote, messages. That's an oversimplification. But a lot of drugs work by increasing the time that a neurotransmitter can spend in the synapse, thus modulating the message that the nervous system can send. So neural integration is the term used to describe the ability to process, store, and recall information to make a decision. So these chemical synapses allow for decision making. They're allowing, do we send another message along the synapse, or do we stop this message from being sent? And our neurons are incredibly well connected and allow for a lot of complex neural integration. So some cells of the cerebral cortex have up to 40,000 contacts with other neurons. So the cerebral cortex is where our consciousness lies. So we make a lot of decisions and make associations between our memories and with our learning. And then our neurons decide whether or not to send the message to the next place in the brain. So the postsynaptic potential is what neural integration is based on. So neural integration is based on the postsynaptic potentials that happen at the receiving cell. So a neural integration, right? Neural integration is the overall sum of all of the signals received on a postsynaptic membrane. So that sounds a little confusing, but let's talk about what happens at a postsynaptic cell. So for a cell to fire an action potential, it has to have at least be depolarized to negative 55 millivolts. So it has to be excited to a threshold level of at least negative 55 millivolts. So there are two types of signals that can be sent to a postsynaptic cell. It can either be excitatory or inhibitory. An excitatory postsynaptic potential, or EPSP, is any voltage change caused by a neurotransmitter or another mechanism that's causing a voltage change increase toward the threshold. So an EPSP is a change that's increasing the depolarization, right? You're getting closer to the threshold. So like an excitatory acetylcholine molecule, for example, when that interacts with the postsynaptic membrane, we're increasing the likelihood of an action potential firing. So that's an EPSP. So an EPSP usually results from sodium flowing inside of the cell, thus depolarizing it. So that's what's being shown here. In response to some kind of stimulus, some kind of neurotransmitter, say, we're increasing the flow of ions inside of the postsynaptic cell, getting closer to the threshold. So we might need multiple firings, multiple releases of different neurotransmitters to get closer to threshold for the action potential to then fire in this postsynaptic cell. Other chemical messages inhibit the postsynaptic cell by hyperpolarizing it, by making it more negative, so it's less likely to reach threshold. So an IPSP, an inhibitory postsynaptic potential, occurs when the cell's voltage becomes more negative than it is at rest. So that's an example of a GABA neurotransmitter stimulus, right? It's allowing the flow of chloride ions in, which is going to make it hyperpolarized and thus less likely to fire. So IPSPs can result from either chloride entering or potassium exiting. So if potassium leaves, we're then still making the inside of the cell more negative. And it's important to know that one neuron is receiving input from thousands of other neurons. So some incoming nerve fibers might produce EPSPs, while others might produce IPSPs. So the overall response of the neuron depends on whether the net input is excitatory or inhibitory. So summation is the term, it's the process of adding up all of the EPSPs and IPSPs and responding to their net effect. So the postsynaptic neuron does summation, right? It does neural integration and summation. It combines all the postsynaptic potentials, right? And this balance between EPSPs and IPSPs will enable the nervous system to make a decision. And please note that EPSPs and IPSPs do not determine whether or not a neuron fires. They only change the rate of firing by either stimulating or inhibiting the production of more action potentials. So the nervous system will already stimulate an action potential from occurring the first time. So a neuron will be fired regardless. An EPSP or IPSP will then change the rate at which that neuron fires by either increasing more action potentials or decreasing more action potentials. So a single depolarization caused by an EPSP is never enough to reach threshold. It has to be the summation of multiple EPSPs that is enough to reach threshold to cause an action potential. And neural coding is the way that the nervous system converts information into a meaningful pattern of action potentials. So there's two types of information that the nervous system has to convert in neural coding. There's qualitative information and quantitative information. So qualitative information depends on which neurons fire, meaning we have different neurons that are stimulated by different types of stimuli. So each sensory nerve fiber to the brain leads from a specific type of receptor that recognizes a specific stimulus type. So what this means is our specific optic nerve only knows how to recognize light. The optic nerve fires in response to light. It doesn't respond to smell or touch. So the qualitative information, right, that light, right, the type of information is light, that will only stimulate the optic nerve. It will not be able to stimulate our motor neurons. Right, our motor neurons are not activated by light. So qualitative information depends on which neurons fire. So when our optic nerve fires, our brain interprets that as a light change. It's the change in light. Right? So the nervous system converts qualitative information depending on which neurons are firing. Whereas quantitative information depends on how intensely or how frequently a neuron fires. So this quantitative information is about how intensely or how frequently a neuron fires. So basically, our optic nerve can either be stimulated very frequently, very intensely, if it's very strong light, or it could be stimulated very weakly, if it's a very weak light. So the qualitative information talks about, okay, what type of information are we dealing with? Are we talking about light? Are we talking about smell? Are we talking about touch? The quantitative information talks about how strong is the light, and how strong the light is perceived is determined by how many times, how many action potentials are being fired by the optic nerve. So I hope that makes sense. And this is the last thing I want to show you, I believe. This is a graph showing action potentials when a weight was applied to the frog muscles. So they took a gastrocnemius from a frog, a leg muscle, and they applied different weights, and they attached a transducer to measure the action potentials. And what you can see is that when they applied a light weight of only 2 grams, there were very few action potentials that were recorded. As the weight increased, the frequency of action potentials increased, right? So 20 grams, the heaviest weight, showed the most action potentials. the firing strength increased and the frequency increased. So the motor neuron is responding. The sensory neuron is responding to the weight, and it is applying the weight is changing the frequency of action potentials that will be sent to the brain. Right? This actual example is not linked to the brain, but if this gastrocnemius were linked to the brain, it would tell us how much weight is being applied. So it would give us an idea of how much force is being applied to the leg based on how many action potentials are being fired in the gastrocnemius. Since a gastrocnemius receptors are of a specific type, that's how my brain knows, okay, this is a weight pulling on my gastrocnemius and not light hitting my retina. So this crash course video should summarize everything for you about the synapses. And if you're interested in any of the extra material on memory or nervous system disorders, please feel free to read on some of the slides. I made them easy to break down so you can teach them to yourself on your own, but do keep in mind that they will not be on the exam. So this is the end of chapter 12, and the next lecture will be on chapter 14, which is the brain. So we're skipping chapter 13, and that will be covered in the next in the lecture that follows the brain. So thank you very much, and this is the end of chapter 12. I hope you have a great day.